Hi, everyone. My name is Eaperkowski. I am a graduate student at Texas Tech University. And today I want to talk to everyone about the impacts of soil nitrogen fertilization and light availability on plant carbon costs to acquire nitrogen in two species with different nitrogen acquisition strategies. So we know that plant nitrogen acquisition is a process in terrestrial systems where carbon and nitrogen are tightly coupled. Right? Plants draw carbon dioxide in from the atmosphere and convert this to simple sugars, which supports net primary productivity and allows plants to allocate carbon towards two primary pathways. First, referenced here as C growth, plants might use carbon to support whole plant growth. This might be through woody tissue production, leaf production and maintenance, or towards storage. Alternatively, noted here as C transfer, plants might use carbon to support soil resource uptake. This can be either through fine root production, root exudation, or carbon exchange with microbial symbionts, such as mycorrhizal fungi or symbiotic nitrogen-fixing bacteria. And plants use this carbon that gets allocated below ground to acquire important soil resources that support photosynthetic and metabolic demand. While there are a ton of different nutrients that plants can acquire, plants acquire nitrogen in the largest quantity. And once acquired, nitrogen can be used for things like the maintenance of structural tissue, production and maintenance of photosynthetic enzymes such as rubisco, and many other important functions that support or that are critical for the growth, reproduction, and survival of an individual plant. However, plants are not able to acquire nitrogen without first allocating carbon below ground. Thus, there are inherent carbon costs associated with acquiring nitrogen. And these can include structural carbon costs associated with root production and maintenance, or active carbon costs associated with the exchange of carbon for nitrogen with microbial symbionts. But I think it's important to note that carbon costs to acquire nitrogen are dynamic and therefore are likely to change with changing above ground and below ground environmental conditions. When we look below ground, soil nitrogen availability likely plays an important role in modifying these carbon costs. And this is because increases in soil nitrogen availability don't necessarily influence plant demand to acquire soil resources, and therefore likely would not have a large impact on the magnitude of a plant's below ground carbon investment. Thus, if plants acquire additional nitrogen without a proportional change in below ground carbon investments, then carbon costs to acquire nitrogen would likely decrease with increasing soil nitrogen availability. And in species that acquire most of their nitrogen through direct uptake, this could be indexed through increased per root nitrogen uptake. However, effects of soil nitrogen availability on carbon costs to acquire nitrogen may be context dependent on the species' dominant mode of nitrogen acquisition. And this has been shown in a few modeling studies and meta-analyses in species that associate with different types of mycorrhizal fungi, species that associate with nitrogen-fixing bacteria, and species that acquire nitrogen mostly via direct uptake. So this figure describes the hypothesized relationship between soil nitrogen availability, represented here on the x-axis, and carbon costs to acquire nitrogen, represented on the y-axis. And species that associate with nitrogen-fixing bacteria are represented here by a yellow trend line, and species that acquire nitrogen through direct uptake are represented through a green trend line. And when we plot these hypothesized relationships, you can see that species that associate with nitrogen-fixing bacteria have lower carbon costs to acquire nitrogen than species that acquire nitrogen through direct uptake when soil nitrogen is less available. This might pose a competitive advantage to species that associate with nitrogen-fixing bacteria under low soil nitrogen environments. And this is because species would be able to acquire more nitrogen from a carbon investment than a species with higher carbon costs to acquire nitrogen. However, as soil nitrogen availability increases, species that acquire nitrogen through direct uptake experience decreased carbon costs to acquire nitrogen. And eventually, you can see that this might lead to a scenario where it's more advantageous for species to acquire nitrogen from direct uptake pathways than it is through nitrogen fixation. And this could potentially cause nitrogen fixing species to switch away from nitrogen fixation and towards direct uptake to maximize nitrogen uptake from a given below ground carbon investment. And finally, above ground environmental conditions might also influence carbon costs to acquire nitrogen. Specifically, environmental conditions that increase net primary productivity and increase plant demand to acquire soil resources should increase carbon costs to acquire nitrogen over short temporal scales. And these patterns would likely be indexed through larger below ground carbon investments relative to nitrogen uptake. For example, if species are growing under environmental conditions where there's increased light availability, we might expect that net primary productivity would increase, which should increase below ground carbon investments in order to acquire additional nitrogen to support higher rates of net primary productivity. So in the context of the last couple of slides and potential sources of variation in carbon costs to acquire nitrogen, we were interested in understanding how soil nitrogen availability and light availability influence plant carbon costs to acquire nitrogen. To do this, we set up a greenhouse experiment where we grew Gossypium hirsutum, which I'll reference from this point forward as cotton, and Glycine max, which I'll reference from this point forward as soybean, under 4 light levels and 4 soil nitrogen levels in a full factorial random block greenhouse experiment. In this experiment, we created the different light treatments using shade cloth, and different nitrogen fertilization treatments were created using a modified Hoagland solution. And I think it's also important to reference here that cotton is a woody perennial species capable of acquiring nitrogen via direct uptake or via associations with arbuscular mycorrhizal fungi, while soybean is a herbaceous annual species capable of acquiring nitrogen through direct uptake or through associations with nitrogen fixing bacteria. We quantified the carbon cost to acquire nitrogen for each individual as a function of root carbon biomass and whole plant nitrogen biomass. There are several limitations that are associated with this metric that I don't have time to get into here, but I will say that if you want more information about this metric, I've linked a QR code at the end of this talk to our recently accepted manuscript on this work that more explicitly details these limitations. We used this experiment to test the following hypotheses. We expected that light availability would impact carbon costs to acquire nitrogen similarly in both species, and that this would be indexed by a larger increase in root carbon biomass than whole plant nitrogen biomass. However, we expected that nitrogen availability would impact carbon costs to acquire nitrogen differently between species. We expected that carbon costs to acquire nitrogen would decrease with increasing soil nitrogen fertilization in cotton, and that this would be due to an increase in per root nitrogen uptake. However, we expected that these carbon costs would not change with soil nitrogen and fertilization in soybean, and we expected that this is because soybean would be able to acquire most of its nitrogen from nitrogen fixation and would therefore be independent of changes in soil nitrogen fertilization. So I want to take a second to orient everyone on these figures before I show the data. From this point forward, all figure x-axes have soil nitrogen fertilization listed as parts per million, and the response variable on the y-axis. On the left panel is cotton, and on the right panel is soybean. And colored points and trend lines represent different light treatments. So we found that nitrogen fertilization generally decreased carbon costs to acquire nitrogen in cotton, and that this pattern was observed regardless of light level. In all cases, we found that this was indeed the result of a larger increase in whole plant nitrogen biomass than root carbon biomass. Additionally, we found that light availability increased carbon costs to acquire nitrogen. And in all cases, this was indexed by a larger increase in root carbon biomass than whole plant nitrogen biomass. Interestingly, though, we found the same patterns to be true in soybean, where increasing nitrogen availability generally decreased carbon costs to acquire nitrogen, and increased light availability generally increased carbon costs to acquire nitrogen as well. And these patterns didn't necessarily follow from our original hypotheses about the impacts of nitrogen fertilization on soybean carbon costs to acquire nitrogen. However, these patterns make a bit more sense when we look at the root nodulation data. We found that, and this is not terribly novel, by the way, but we found that increasing soil nitrogen fertilization decreased root nodule biomass, and that this is a pattern that was observed across all light treatments. And we think that this likely decreased the nitrogen fixation capacity of soybean individuals. However, what's interesting about this is that this occurred alongside an increase in whole plant nitrogen biomass with increasing nitrogen fertilization. And this suggests that soybean individuals growing under high levels of soil nitrogen might have been accessing nitrogen from an alternative source aside from nitrogen fixation. And when we look at the ratio of root nodule mass to root biomass, we see that root nodule biomass is decreasing relative to root biomass, regardless of light level. And so these patterns do suggest that individuals may have switched their dominant mode of nitrogen acquisition away from nitrogen fixation towards direct uptake when soil nitrogen became more available. So to put our findings together, we found that light availability increased carbon costs to acquire nitrogen in both species, which supported our hypothesis. We also found that nitrogen fertilization decreased carbon costs to acquire nitrogen in cotton, which also supported our hypothesis. However, we observed similar patterns in soybean, which did not support our hypothesis, where, again, our root nodulation data suggests that these patterns were likely due to a shift in the dominant mode of nitrogen acquisition away from nitrogen fixation to direct nitrogen uptake. So I think it's important to note here that we did not compare carbon costs to acquire nitrogen between species. This is because cotton and soybean are not phylogenetically related. And because we used one nitrogen fixing species and one non-nitrogen fixing species, we are extremely limited in our ability to extrapolate these patterns to all nitrogen fixing or non-nitrogen fixing species. So a simple solution for this is for future work to use a larger range of taxonomically related species to determine how generalizable these patterns are. Additionally, we conducted this study over a short 5-week temporal scale. So there's also a need to evaluate these patterns across other spatiotemporal scales to determine the range for which these patterns hold. And finally, we would love to evaluate the threshold of soil nitrogen availability where nitrogen fixing species might shift away from nitrogen fixation towards direct uptake, and compare these results to patterns that are simulated in Earth system models. And finally, I'm running a bit short for time, but this work would not have been possible without the help of several undergraduate researchers. I'd also like to thank members of the Schwilk, Van Gestel, and Johnson Lab for feedback on both this presentation and for feedback from the manuscript that came from this. And we'd also like to thank Dr. Paxton Payton at the USDA station in Lubbock for providing us with cotton and soybean seeds that were necessary for this experiment. And finally, for those that are interested, this work is part of a recently accepted manuscript at the Journal of Experimental Botany. And so if you scan the QR code below with your phone, you'll be able to access the manuscript landing page. And with that, I just want to say thank you, and I'd be happy to take any questions